evening all and very warm welcome to the session. So really very happy to meet you all in this evening. So this is going to be a molecular biology top 5 concepts that you should never miss whenever you are preparing for your examination. Yes. So your CSIR examination forms are out. So everybody maybe know you could have come across that these forms are really out, and please take time to fill the form, and please keep yourself reminded that always the forms it should be properly filled. Okay, so don't miss out any particular points. Okay, so make sure that you are going to fill the form correctly. I hope that I am visible, I am audible and all. Yes, everybody. So I am visible, I am audible. Am I completely available to you all? I hope that this is all clear. Okay, fantastic everybody. So good evening all. So this is Dr. Saranya. I am talking to you about this most important topics with your CSIR NET molecular biology. So we are going to revise molecular biology. This is a little bit about me, if at all you are watching me for the first time. Right, so I have completed my Ph.D. in Biomaterials. So I hail from Unacademy University and I have already cracked the examination, and I have 15 papers to my credit, and I am teaching in Unacademy for a long time, since 3 years, and already I have 10 plus years of teaching experience. With this I am seriously helping you all to make your examination to be prepared in a better manner. So this is regarding this Unacademy Plus and Iconic subscription. So basically what is the difference between the Plus and the Iconic subscription is that in the Plus you will have normal features like you can attend the live classes, you can go for this weekly test, and you will have all the options that you generally need for your preparation. This Iconic is little bit extra. You will get test analysis, you will get expert guidance, you will have a personal guidance and a planner. Soon the study materials will be released, so you will not have to worry if you take up with the Iconic subscription. Okay, so these are the benefits of the Plus and this Iconic subscription. Okay, so if at all you have not yet subscribed, it's high time that you have to come up with a subscription. Absolutely, you can use my code Saranya Live. Okay, right. So I'm going to teach you with unit number 1, that is your biochemistry, unit number 2 cell biology, unit number 3 that is going to be your molecular biology, unit number 5 developmental biology, unit number 10 ecology, unit 11 evolution, unit 12 applied, and 13 methods. Okay, so these are the units that I am going to take up. So seriously it will be of great help to you. So follow me in our official Telegram channel and enroll yourself with the special classes. Okay, so this is what we are going to see today. Yes, I told you that we are going to see the top 5 what is that? Top 5 topics of your molecular biology. Right, you can see that in this few have a red dot and few it is not having red dot. So what is this red dot telling about? This red dot is representing the methylation. Okay, so if at all this methylation is taking place, okay, so if at all this methylation is taking place, okay, absolutely it is going to, yes Preeti, good evening, yes Priyadarshini, very very good evening. So if at all this methylation is taking place, it is absolutely ready for replication. Okay, so if it is going to be in a hemimethylated condition. So what do you mean by hemimethylated condition? You can see here, one it is methylated and the other one it is not methylated. So this is what is called as a hemimethylated condition. So if at all it is going to present in a hemimethylated condition, there is one fellow, yes Plati, there is one fellow who is going to inhibit. Do you know who is that? Who is that fellow who is inhibiting whenever your DNA it is going to be hemimethylated? His name is going to be CA. Okay, so if at all your DNA is hemi means partly methylated, this CA protein is going to come and it is going to bind your origin. So now can your DNA replicate? Not at all possible, isn't it? Right. So the CA, it is going to serve as an inhibitor. Okay, so what the CA is going to do? It is going to inhibit your replication. Only fully methylated origins can undergo replication. If at all it is hemi-methylated, gone forever, it cannot undergo replication. Okay, right. And what is this protein which is actually initiating your replication? It is called as, what is that name? DNA A. Okay. So in prokaryotes you call them as, DNA A. Okay, right. And in eukaryotes, how do you call them as? In prokaryotes, you call them as DNA A. In eukaryotes, do you know the name? This is what it is present in your licensing factor. This is nothing but it is going to be your CDT1. So who is that? It is going to be your CDT1. Okay, CDT1, it is the name that is being given in that of the eukaryote. Okay, so in prokaryote you will call them as your DNA A. Okay, right. This DNA A, very good Priya. So this DNA A, it is going to be a ATP dependent molecule. Okay, so this DNA A, it is always going to be an ATP dependent molecule. If ATP is involved, absolutely there is going to be the hydrolysis. Whenever there is going to be this hydrolysis, it is going to tell you something. What it is going to tell you? Whenever there is going to be this hydrolysis, yes, it is going to kick away something. It is going to send away something. Yes, Tasneem, it is going to send away something. What it is going to send away? It is going to break up your bond. That is what is called as DNA unbinding element. What is that? DNA unbinding element. Okay, so this happens exactly near your origin. Okay, right, in your origin you will contain this 9 mer and 13 mer. Okay, right. Have you ever used this bags, maybe biohazard bags, even your normal dustbin cover bags? So how do you open them? Yes, have you ever tried to open it? See, this is how the bag will look like. Okay, right. So both of the ends will be closed, right? So you will just rub near the ends. If you rub near the ends, what will happen to that? Here it will bulge up and open. Have you ever noticed? In the end, if you just roll up, it will bulge up and open. The same process it was or it is happening with your replication. In the replication, whenever you want to open up your DNA, you are going to put pressure in the nearby region. If you put pressure here, this region is going to open up. Understood the aspect? Everybody in this class? Yes, this DNA A, it is going to associate with your 13 mer. This is what is going to be your 13 mer. Since because it is associating with the 13 mer, who is opening? This 9 mer is begin to melt. This 9 mer is begin to open up. Do you understand how it works? Exactly like your dustbin car or like your biohazard bag. You want to open up the DNA, you put the pressure in the nearby region. The moment you put the pressure in the nearby region, your bag is opening. Likewise here, along with this ATP, your DNA A, it is going to interact with your origin. Where it is interacting with your origin? It is interacting exactly in the 13 mer region. So where it is opening? It is opening in the 9 mer region. So this 9 mer region is beginning to open up. Okay, so don't think that helicase is only doing this unwinding. Helicase unwinds, but it needs an initiator. Without melting, unwinding can never happen. Can you imagine where this melting can take place? I told you a clue, it is in the origin. In the origin, where it is going to do? It is going to do exactly at the AT rich element. Okay, so in this AT rich sequence only, it is going to melt. That too it is located within the 9-mer region. Yes, Priya, in the AT rich region it is going to melt, and that too it is in the 9-mer region. Once your DNA comes, it is going to create a ATP. So this ATP hydrolysis is going to give the energy. Once the energy comes, your DNA, it is going to get opened up. Once your DNA is open, helicase can come and unbind. Everybody in this class is clear about this aspect. And I told you another important protein. What was that? CK. So what the CK will do? What the CK will do? The CK, it is going to inhibit the hemimethylated DNA to undergo the replication. Okay, so the CK, it will allow only the, what the CK will allow? The CK will allow only the fully methylated DNA to undergo replication. If it is going to be hemimethylated, CK will go and bind with this hemimethylated. So your replication is going to be inhibited. Right. So these two proteins are now clear. DNA A, CK. These two are the most important protein. Now we are going to learn the third important protein. Concept number 1 only we are doing. Yes, we are doing with replication. Concept number 1 today. Yes, right. So the third important protein that you should never miss, it is going to be the HEDA. Okay. So what is this HEDA doing? It is helping or it is inhibiting? Yes, I told you that if I take my CK, okay, number 1 I told you about the CK, it is going to inhibit, I told you, because it is going to inhibit whom? CK will inhibit hemimethylated DNA from replication. Okay, if CK is binding means replication is inhibited. Okay, number 2, it is going to be your DNA A. DNA A, it is the name that is being given to this prokaryote. Whenever DNA A is bound, it is going to melt. Okay, it is an ATP associated protein, so it is going to melt your bases, that too at the 9-mer, and it is going to bind at the 13-mer. That is what it is happening with the DNA A. Right? Fine. Third one I was telling you is about this HEDA. So what this HEDA will do? So what this HEDA will do? The HEDA, it is only helping in giving this ATP to your DNA A. Got the point? Actually, while incoming, the HEDA will come as ATP HEDA. Okay, the HEDA is only bringing the ATP. But when your DNA A comes, okay, this HEDA is going to provide with an PI. Okay, right, and it is going to help to mobilize your DNA A. Okay, so this DNA A, it is making use of this ATP. So who is going to give away this ATP? This is going to be your HEDA. So 3 major proteins we have done today. So shall we make a quick recap? Everybody in this class, right? You are going to tell me whether it is true or false. Okay, number 1. Yes, number 1, it is the CK. CK, it is an inhibitor of replication. True or false? Everybody in the class should answer. CK, it is an inhibitor of replication. True or false? Come on guys, everybody can make it out. Make it fast, children. Yes. Come on guys, everybody can do the simple question. Very good. Priya says it is true. 100% correct, Priya. So this is going to be a yes, Mridhi. 100% correct. It is a true statement. Why? Because CK, it is going to inhibit the hemimethylated DNA. If your DNA is completely methylated, CK can never bind. Okay. So CK cannot bind your yes, Smriti. It cannot bind your fully methylated DNA. It cannot bind. CK will bind only the heavy methylated DNA. Okay. HEDA, it is going to help the DNA for initiating replication. True or false? HEDA protein, it is helping in this DNA initiation. True or false? Everybody in this class, think and answer me. Header, it is going to help the DNA for initiating the replication. It is going to be a true statement or false statement. Yes, Smriti, Priya, absolutely correct. It is going to help your DNA. How? By providing your ATP. Okay, right. DNA, it is going to bind 9mer. True or false? Think and answer. DNA, it is going to bind the 9mer. True or false? It is a true statement or false statement? DNA. DNA, where it will bind? It will bind to the 9mer or 13mer? It had to bind with your 13mer. Yes, Kavya, very good. It should bind only with the 13mer. But this DNA will melt your 9mer. Okay. I told you like your dustbin cover. Okay. So it is going to bind with the 13mer and it is going to melt your 9mer because 9mer is only going to be rich in your AT region. Understood? Right? So these three proteins are thorough to you. Okay, fine. So there is a very much similarity in mechanism between your replication that occurs in your prokaryotes and eukaryotes, apart from major differences in the type. Yes, Priya. So this header, this is a very important protein. What this header will do is that it is going to help in your ATP hydrolysis. Okay, right. This header, okay, so it will come as your ADP header. Okay, so your DNA, it is undergoing your hydrolysis and it is going to liberate this PI. No, so in this process only, this header is going to render a help. It will help in this ATP hydrolysis. Because of this ATP hydrolysis only, your DNA, it is getting the energy of PI. Okay, right. So once this is getting this PI, it can open up your DNA. For this, you require this hydrolysis. No, for that, who is helping? This header is helping. Got it? Okay, fantastic. So, and this is an equivalent of your prokaryotic and eukaryotic fellows. Okay, so let us see what are all the equivalent things in your prokaryotes and eukaryotes. Okay, fantastic. So, in your prokaryotes, you call them as DNA B. So, what is this DNA B? It is nothing but it is going to be the helicase. So, what is that? It is going to be nothing but your helicase. In your eukaryote, you call them as MCM. When it will be recruited? Yes, everybody. So, when it will be recruited? MCM in the G1 phase or S phase? MCM is recruited in the G1 phase. Okay. I told you, no, you will study before the examination. But you have to write the examination only during the examination time. Okay, even though you prepare so much before the exam, you cannot write your examination before. Only at the exam time you can give the exam. Likewise, this helicases will be loaded before the synthesis, but your initiator, who is the initiator? DNA A in prokaryotes and then your CDT1 in your eukaryotes. It is a licensing factor that will be recruited only in your S phase. It can never be recruited earlier. Only in the S phase it is going to be recruited. Got it? Right. Fine. So the next is about the helicases. I told you helicases is equivalent of DNA B in prokaryotes. In eukaryotes you call them as MCM. So this helicase should be kept silent, isn't it? It should be kept quiet. So who is going to keep this quiet? It is going to be the chaperone. So who is making him silent? It is going to be the chaperone. Chaperone is like a folder, isn't it? Right. So it is going to be kept quiet. So this is what is done by your DNA C in your prokaryotes. In your eukaryotes it is called as CDC6. So this is also recruited along with your MCM. else your MCM will become active now. So MCM should be inactive till your S phase. So this is also recruited. So during your licensing factor or during your license time, this will be removed. This will be sent out. So once this is being sent out, now your helicase is free. Now already by this time your CDT1 has come. So your DNA has been melt. So once your DNA is melt, the total process is going to take place. Understood this wonderful story of how this unwinding and how your DNA A is helping out in this process? Everybody in this class, did you get it? Shall I ask you a couple of questions before we move out to the next concept? Yes? Okay. Question number 1. MCM, it is recruited in S phase only. True or false? MCM, it is recruited only in the S phase. True or false? Absolutely, this is going to be a wrong answer. What should have been correct answer? It should have been in the G1 phase, not the S phase, but it should be in the G1 phase. Very good. Right. Second one. DNA A, it is recruited in the G1 phase. True or false? DNA A, it is recruited in the G1 phase. True or false? This is a false statement, isn't it? Why? DNA A should come only in the S phase, not before. Okay, right. CDT1 is equivalent of your DNA B. True or false? CDT1, it is an equivalent of your DNA B. True or false? Absolutely, it is going to be a false statement. CDT1, it is going to be DNA A. It is an initiator. Okay, correct. MCM is only going to be your DNA B. And what is your DNA C? It is the CDC6. CDC6 is going to be your loader, your helicase loader you can say, or you can call it as a chaperone also. It is going to be fine. Alright, fine. Yes. Okay. Next, a single stranded is there means it will be stabilized by single stranded binding protein. Okay, so it is what is called as your RPA in case of the eukaryote. In eukaryote you call them as RPA. So who is going to do this priming job? Primase is there, no? Yes, Somnath. So as the primase is there, no? In case of your prokaryotes, you call them as DNA G. Okay, so DNA G is only doing the primase. Fine. So do RNA require a primer? Anywhere you have studied that RNA, it is requiring a primer? No? Yes, Amrita. RNA never requires a primer, isn't it? Only your DNA requires a primer. Why your RNA is not requiring a primer? It will begin the synthesis because the bond between the two ribonucleotides are very, very naturally formed. So you don't require a primer for the 3' OH end. Okay, so this 3' OH end is free. It can be incorporated easily in case of RNA. But if at all you are working with your DNA, my goodness, you cannot have a free 3' OH. So because of this 3' OH only you require a primer. So this primer is going to be your primase. Your primase is nothing but your RNA polymerase, because RNA polymerase is only going to synthesize your RNA primer. Got the logic now? Yes? Okay, so what is the nature of the DNA G? It is an RNA polymerase. Okay, what is the direction of this RNA polymerase? Think and tell me what should be the direction of my RNA polymerase. Can this RNA polymerase move in opposite direction? No. Always it should be 5' to 3'. The same with your DNA polymerase. This is also 5' to 3'. Okay, so what you will do in this Okazaki fragment? Okazaki fragment, is this direction going to be 3 prime to 5 prime? Not at all. Any time, whichever is the strand, let it be your leading strand, let it be your lagging strand, always remember the direction of DNA or the RNA synthesis, it will take place only from the 5 prime to 3 prime. Got it? So now everybody is clear in this? Okay. So we saw a sliding clamp, no? Yes? Right. So who is this clamp? What is this clamp made up of? Yes or not? So what is going to be this clamp made up of? This clamp is made up of beta in case of your prokaryote, and it is called as pCNA, polynuclear. Okay, so that is what is pCNA. So this pCNA, it is what is going to be present in case of the eukaryote. This is going to be a clamp. Clamp is what will hold everything together. In the clamp only, who will be sitting? Your polymerase will be sitting. Right. So for this polymerase to sit, you need a loader. No. The loader is only going to be your gamma delta complex in prokaryote, and it is going to be RFC in case of your eukaryote. Now you got it? Gamma delta in case of your prokaryote and RFC it is in case of eukaryote. And who is carrying out this replication? It is by replicase or pol 3 in prokaryotes and by delta and epsilon in eukaryotes. So who is synthesizing the leading strand? E is there, no? Epsilon. Who is synthesizing the lagging strand? It is delta. Okay. So technically speaking, leading strand is 5 prime to 3 prime, lagging strand is 3 prime to 5 prime. But I told you, no polymerases can move in the opposite direction. That is why in Okazaki you are producing them in the fragments. So it will be produced in the loops. Later this will be connected by your ligases. Got it? So now you understood the total process of replication, of how it occurs. So there are variety of types of your replicase polymerases. So your alpha, it is for nuclear replication, delta for lagging, epsilon for leading, and then gamma for mitochondria. Apart from that, you have the low fidelity enzymes, tau for base damage, neta for thymine dimer, K for deletion of bases. So all these things are seriously very important because it is all going to make up your replication. So this is in eukaryotes. In prokaryotes, what do you have? In case of prokaryotes, you have your DNA polymerases. The pol 3 is the replicase. It is only helpful in the replication. Pol 1. So pol 1, it is encoded by this pol A. Okay. So it is only helpful. This pol A, it is only going to be helpful in many purpose. It is helpful in repairing your damaged DNA. This damaged DNA repair is there, no? That it will do. Also it will help in this primer removal also. Okay, so this primer removal, it is also done by this. Nick translation is also done by this. This is only making your clino fragment. So all this is coming under your pol 1. So pol 1 is making all these things. Pol 2, it will restart the replication, and then pol 3 is your actual replicase. Apart from that, pol 4 and 5 are error prone polymerases. So they do not check. There is no proofreading. It is all error prone. What is this diagram about? Can you guess? Something at the end it is happening. No. So this is going to be the telomerase. What is this telomerase? It is an RNA dependent. See, this is an RNA template. No. So usually for your replication, what is the template? Yes. For the replication, what is the template? For your replication, what will be your template? Yes, everybody think and answer. For replication, what is the template? Template should be your DNA, no? Yes, DNA will be the template. I told you that majority of the time 3 prime to 5 prime is the template for your leading strand because this fellow can move from 5 prime to 3 prime. No, your enzyme. So 3 prime to 5 prime is going to be your template. It is a DNA. But here for your telomerase, who is the template? It is an RNA template. So that is why your telomerase, it is called as RNA dependent DNA polymerase. What is the telomerase called as? Telomerase is called as the RNA dependent DNA polymerase. Is it all clear to you? Yes? Okay, and there are several modes of replication. Okay, so it is going to include the semi-conservative, so both will be having one hybrid. Okay, so in only semi-conservative you see a hybrid, none other you will see a hybrid. Okay, so only under the semi-conservation you can see a hybrid. Okay, so each from one parent, it is called as semi-conservative. And if it is going to be conservative, no mixing up. In dispersive, throughout it is mixed up. All the 4 strands will contain all the components. It is called as dispersive. Okay, right. So your replication can go in your theta model, rolling circle model, or your this direction. Okay, right. So this is all about the replication. Okay, so are you all clear in replication? Everybody in this class? No more doubts in this replication? Do you remember all these terms? Yes? Yes, Priya. So why your RNA does not need a primer? Okay, right. So very good question. In case of this RNA, okay, so especially if you take up this RNA, okay, right. So let me tell you, this RNA, it is going to have your ribonucleotide bond. Okay, so since because your RNA, okay, it is going to want a ribonucleotide bond. Okay, it is not at all requiring your primer. It can directly add your first thing. Okay, right, because this 2 prime OH will stabilize your 3 prime OH. So even if your first DRTP is coming, see, you have your DNTP. Okay, deoxyribo nucleotide triphosphate protein. You have your RTP. Okay, right. So this is little bit unstable. Why? Because this 2 prime OH is not there. Okay, but here the 2 prime OH is there, no. So it is stable. So easily it can form a bond with your it will make your 3 prime OH to make a bond with your 5 prime phosphate. But this one, if it is going to be alone, there is no 2 prime OH, no. So if this 3 prime OH is going to be alone, okay, it cannot form a first bond, but it can extend a bond. Okay, so there will be few, no. If something new comes in the market, okay, you will not buy something new. But if your friend buys, okay, and the friend is suggesting you, you will think, okay, my friend has used, so this is going to be better. So it will extend. That is what your the DNA polymerase is doing. If a primer is there, it can extend, but it will not create a new bond. But your RNA polymerase is not like that. It is creating even the initial one. Okay, so that is the major difference between your DNA polymerase as well as the RNA polymerase. Yes, everybody, I hope that this is all clear to you. Yes? Okay, fantastic guys. So please do come up with the subscription. Yes, this month I am doing a lot of things on methods. Okay, and also many revision courses are there on board. So never miss out. Yes, so whatever you have learnt, please try to revise. Please be regular to the class such that definitely you can gain a lot of knowledge. All the very best guys. I'll keep you up meeting in another important session. Take care till we meet. Thanks a lot everybody. Thank